My student Addy and I posted a preprint online on Lamiales. The link is in the description below. I like to chat about it because one of the reasons I'm making these videos is that the PGR-TK plots are quite dynamic, and when you click on a part of the sequence, the software highlights the conserved segments in all other sequences, and that's quite helpful to explore the plastic sequence conservation. And especially for inverted repeat, the software highlights both segments of the repeats, and this aspect cannot be demonstrated by the static images in the paper, so I thought a video would be helpful. Now, Lamiales is the mint order. It's the large order with about 24,000 species split into 24 to 26 families. The largest family is Lamiaceae, which is the mint family. It includes basil, mint, roseberry, sage, oregano, thyme, all of these herbs that you grow in the garden, and includes lavender. And you can see that they all have nice smell. But in addition, Lamiales also has a lot of interesting plants. So one family includes bladderwort. It's a large genus with carnivorous plants. So that's one of the interesting ones. And then also a separate family includes orobunch, which is a parasitic plant. And there are a number of other parasitic plants in that family. Another very interesting plant that I have seen is a desiccation tolerant plant called resurrection plant. When you keep this plant without water, it can live for many months, but as soon as you give water to that dried plant, it will start growing again. So overall, Lamiales is a colorful order, both literally and figuratively, and I'll go over what I see in terms of the evolutionary patterns on the plastid data. I already posted a video on this PGR-TK software. It's an entire video on what the software does, some of the common patterns, and I'll not repeat the content. Overall, what this PGR-TK software does is it takes a group of plastid sequences, compares them, finds the conserved patterns, and then demonstrate the pattern nicely in a plot. And this plot is interactive, so you can also identify patterns of conservation. And in the previous paper, I talked about a number of patterns in hundreds of PGR-TK plots that we analyzed, and Lamiales is no different. As I go over these sequences, I'll show you those patterns in the context of Lamiales. In the next part of the discussion, I'll show you a number of PGR-TK plots of different Lamiales genera and families. First, I'll show you what is expected and then discuss what is unexpected. So let's start with the mint or mentha genus. Here is a PGR-TK plot. What you are seeing here is a set of number of plastid sequences. Each horizontal line here is a plastid sequence. What PGR-TK does is compare between those sequences and then highlight the conserved parts of those sequences. On this end, you can see the genus and species names of those species. And then here you can click on any of the segment and PGR-TK will highlight all of the identical blocks. Let me show you. So I clicked here and you can see that the entire group of sequences are highlighted. One thing I did before making these plots is to align those sequences in a way so that the identical blocks are all on top of each other. But in general, this feature is quite helpful when these sequences are not nicely aligned. You may see a concept block which is here in the first sequence, but here in the second sequence. Remember, the plastids are all circular. So even if I move these and do a circular rotation, they will still be the same plastid sequence. Next, let me show you on this plot by highlighting this segment. I'm going to click on this blue segment, and you can notice that two blocks are picked by the software. Why is that? Because this is a part of the inverted repeat. What inverted repeat means is here this sequence on the forward strand will be identical to this sequence in the reverse strand. Now, if you see the arrows, the PGR-TK software shows arrows. So here it is pointing towards the left, and here it is pointing towards the right. And that's how the software shows inverted repeats. If they are simple repeats, they will be both pointing in the same direction. I'm going to pick the rest of the inverted repeats. So here and here. Now you can see that these three segments are parts of the inverted repeat. This is followed by this sequence in the beginning because the plastids are circular. But now that I highlighted the inverted repeats, you can also easily determine the long and short single copy region. So this entire region in between these inverted repeat is the long single copy region, and this block is the short single copy region. So that's the typical PGR-TK plot that I have seen in most of the plastid sequences. About say 80% of the genera of Lamiales will follow this pattern. Let me show you another one. Here I have Salvia. Takes a minute to load. Okay, so here I have Salvia, and it's very similar to what you have seen in Mentha, except that the sequences are a little more fragmented. One reason for this fragmentation is because the number of sequences being considered is larger. So as you have more sequences, and PGR-TK compares them in pairs, with more sequences there are more variations, and that's why you get a bit more fragmentation. But you can also see that the inverted repeats, like these sequences, this is the part of the inverted repeat, the other end is at the beginning, and this is also inverted repeat. This is also inverted repeat. Let me just highlight them. Another one. Okay, I highlighted most of the larger blocks of the inverted repeat. Also, these portions are inverted repeat, but you can see that this entire sequence here and this entire sequence here going back to the one block in the beginning are parts of the inverted repeat. You can also find that these are the short single copy regions and these are the long single copy regions. One thing you can clearly see is that the inverted repeats are, the blocks are longer, and that means the inverted repeats are better conserved among the plastid sequences than the short and long single copy regions. And that's a pattern that's well known for plastids and it's well recognized. So you can see that the PGR-TK confirms that the inverted repeats are better conserved than the other single copy regions. In the next few plots, I'll show you what the uncommon patterns are. But before I go there, I want to also highlight the scale of the length of the plastid sequences. If you look on the top here, you will find that this is the scale of 160,000 nucleotides, and these sequences are a bit shorter. So typically you will find that most plastid sequences are around 145 to 155,000 nucleotides long, and this is the mint genus, and it's not an exception. Now let me show you a number of PGR-TK plots where the sequences are not so uniform. So I'll start with Incarvilia, which is this one. You can see there are only 4 sequences, but they look quite divergent. You can't really see the inverted repeats. I tried to highlight part of the inverted repeat, which is these segments, but they are quite small. You don't see the nice long conserved inverted repeats. And even though there are only 4 sequences here, they are extremely divergent. You can see how many small fragments are here, and you cannot really highlight any sequence that is conserved or cannot do that easily. Maybe there are these small fragments which are conserved, but they don't show as nicely as the previous figures. The length of these sequences are around 160,000, so somewhat bit longer than the other ones, but in the same ballpark. Now, next one is the Utricularia. It's this carnivorous plant, and you can see that this is a little better than the Incurvia, but you can see the conserved inverted repeats like this blue segment and then these blues and all those. So the inverted repeat is a bit more conserved, but still the sequences are extremely fragmented. There are all these pieces that do not make sense, like these sequences which are only present in uh, a single species, Utricularia aurea, and then the bottom two sequences are a bit shorter, and then also these sequences do not match the rest. So quite a bit of divergence that you can notice here. The next one is Orobunch. Now Orobunch is an interesting case because you can see the length is 130,000 nucleotides, and that's for this long sequence. The rest are below 100,000. So Orobunch happens to be a parasitic plant, and it's well known that in parasitic plants the plastids lose quite a few of the genes because they can use the other plant to get their resources. So they lose many genes, and therefore the plastid sequences shrink quite a bit. And you can see that. But in addition, here you cannot observe any of the inverted repeats or other sequences. In addition, the bottom sequence, that's called Orobunch californica, is quite divergent from the rest of the sequences. In addition, all these sequences look quite different in terms of both length and the sequence content. I'll show you another one. This is a different pattern. Now this is the Plantago, and here the sequences are quite uniform, unlike the ones I have shown you before, but they have two distinct lengths. Here you have a group of shorter sequences, and these shorter ones are of the length of typical plastids, 150 to 160,000 nucleotide, and then they have a bunch of longer sequences which are 165 to almost close to 170,000 nucleotides. That's why the scale goes to 170K here. So this is a different pattern. You see two different lengths within the same genus. Now let me show you another one, Jasminian. Here you have most sequences are of similar length, close to 170,000 except one. And you see quite a bit of fragmentation once again, but the inverted repeats are not missing. You can distinguish the inverted repeats. So it's somewhere in between most the cleanest plots and the incurvalia kind of plots where nothing is very well conserved. Here the sequences are well conserved, but only thing you see is the level of fragmentation is higher than the typical plots. The last one I'll show you in this group is Burleria. Here once again it's a plot that is a bit fragmented, but you can see the inverted repeats, except that this particular sequence, the last one, is shorter. Next, I'll move to the family level plots, and we'll start with the family where Barleria belongs because I wanted to highlight something interesting. So this is the family. So there are way more plots and takes a bit of time to load. But once again, you see all these different genera and species of plastids on this plot. Generally, these sequences of the same genus group together. For example, all these sequences are Justiceas. You can see all these Justicea, except that there is one sequence that got inserted. Now, I wanted to show you the Barleria because you have noticed in the previous plot that these ones have one length, and then the last one, Burleria siamensis, has a different length. Now it looks like the family level plot may give an explanation of why that happens. So here you can see the four Burleria sequences or five sequences which have the same length all grouped together. But where is Burleria siamensis? It shows up here on this, and in terms of length, it matches these Strobilanthes species quite well. So, in addition, if you look at the sequence pattern, it looks like all these sequences, which are mostly Strobilanthes and one Burleria, they all look similar. Whereas here you have the rest of the Burleria, which all group together. PGR-TK also puts together a phylogeny based on the sequence similarity, and you can clearly see that these Burlerias are all grouped together, and then the remaining Burleria joins with the strobilanthes here, strobilanthes. So based on what I see here, I strongly suspect that this particular Burleria sequence, what is given as Burleria, is misclassified. It should be put under the Strobilanthes uh, genus. Now you can see that this is a really powerful software which helps you visually identify the patterns, and based on that determine the genus and species level of all these plants. So I find that quite helpful. In addition, you can see here, you can also highlight the inverted repeats in the family level plots, and both segments show up. And let me click another one, small green one. Okay, so I clicked a few segments of the inverted repeat, which also help you determine the short single copy region and long single copy regions, the entire segment there. And you can see that the entire family has the inverted repeat well conserved because you can highlight them all together. So that's also an indication that these plants are grouped together in the same family correctly. Let me go to new set. So this time I'll pick this one called Lentibularaceae. This has three different genera. One is Genlycia, one is Pinguicula, and Utricularia. Yeah, I've already shown you the Utricularia. Let me show you the genus level plot of Genlycia. Now, if when I click the Genlycia at the genus level, I see that all these six or so plots are quite similar, and the bottom one really stands apart. You can see, except for the inverted repeats where they match generally, but other than that, the last one, which is the Genlycia violacea, is quite different from the rest. Now, the family level plot gives some clues. You can see that these Genlycias are together. These are the Genlycias which are similar. And then Genlycia violacea groups with Pinguicula. And you can see the phylogeny generated by PGR-TK. This is the Genlycia violacea. And once you see here, it may make more sense because this one has stronger similarity with Pinguicula. So we may have to rename this plant or reclassify. And then you can also find the utricularias are all grouping together except the last two I've shown you before were a bit shorter. Now, if you see that phylogeny, the last one is actually an outgroup compared to all of these plants. So this is the Utricularia remiformis, which stands outside the rest of the sequences. And when you looked at the genus level plot, you can see that this remiformis and adpressa look a bit different from the rest of the Utricularia. So there is a lot here. We can go and dig deeper into these sequences, go check why these portions of the sequence are so dissimilar to the rest. So this is just giving you more things to check in those sequences. Maybe they are assembly errors, or maybe the plastic sequences differ quite a bit compared to the rest. So PGR-TK is a nice way to start the analysis. All these calculations take only a minute or two. If you have a small group of plants, say 8 or 10, this analysis takes just 1 minute. So in 1 minute, you get this plot generated. For larger ones, like these uh, family level plots, even this one is not that large. But for say 30 or 40 plants, I have to wait for 10 minutes or 20 minutes, and I get this whole pattern from which I know a lot about how the plastic sequences are conserved. I'll show you two more families, and the reason I'm picking them is because they include some of the genera I showed you before. So this one is Bignoniacea. And the reason I pick it is because it includes Incarvilia. If you remember Incarvilia, it has completely messed up PGR-TK plot. You see a lot of fragmentation. There is nothing well conserved. Even the inverted repeats are quite fragmented. Now, Bignoniacea includes it, but when I look at the entire family level plot, Incarvilia stands quite apart. So these the bottom four sequences here are the uh, members of the Incarvilia genus. And if you see this phylogeny generated by PGR-TK, these four are nearly an outgroup of the entire set. Everything else fall together except these four. In addition, when you look at the sequences, you will find that the sequences have a bit of uniformity in the top part, and only when you check Incarvilia, you see more fragmentation. Although the best way to resolve this is by redoing the plot, by doing the family level plot with and without Incarvilia. I'll try that later, but I just wanted to go over the family level plot as it's generated. Here I highlighted some segments which are conserved between all. So this yellow segment, which I believe is part of the inverted repeat, shows up in all of these plots, including the bottom four, which are Incarvilia. The next family I'll show you is Orobanchaceae. If you remember, I have shown you the plot of Orobanch, which is this one, and it includes all kinds of shorter sequences. This is a family with parasitic plants, and you can see that they are not only shorter, they have all kinds of different lengths. The peak length of the scale here is 130,000 nucleotides, which is only this particular plant, but the rest are less than 100,000 nucleotides or even way shorter. So this is the Orobunch. Now let's take a look at the family level plot where Orobunch belongs. And this is the plot here. This family includes a number of other plants. Uh, this includes Pedicularis, which is another parasitic plant, and then Rehmannia and quite a few different other genera. If you look at the plot, the scale here goes to 200,000 nucleotides because of just one plant, which one plastid sequence. And I'm not even sure whether that plastid sequence is correct or not. So this particular one, Striga asiatica, has much longer sequence. And wait, overall, you see this family level plot looks completely messy. You can maybe in the top part with pedicularis, you see some uniformity, but as you go down, when you go to the Orobanch section, you will find that all these sequences are show no uniform pattern except maybe locally, only a small group of sequences. So this is a really uh, messed up or quite complex sequence, and it will probably need a lot more time to reanalyze, where I would suggest removing some plots and also reanalyzing the sets of plants in um, groups and stuff like that. So this is a really interesting plot in that way. Alright, let me quickly summarize what you have seen here so far. I use this software called PGR-TK to compare the genus level and the family level plastid data from Lamiales. This software compares these sequences, the plastid sequences, and then draws a plot with highlighting all the conserved regions. And based on these highlighted plots, you can see how well conserved the plastids are within a genus or family. And mostly the sequences show the short single copy, long single copy, and inverted repeat regions very clearly, but I've shown you a number of plots where, or number of genera where they don't, and also usually within the genus the plastids have similar length, but I've shown you a number of exceptions. And the third pattern I have shown you is the set of these plastids which are not correctly matching the rest of the sequences within the genus, and most likely that's because these plastid sequences or the plants themselves are from a different genus. They are either misidentified or they have a sequencing error. I don't know what the cause is. And then I have shown you a number of interesting plants from parasitic type or the Utricularias, the carnivorous plant. So overall, Lamiales has a number of patterns, but they match what I have seen from PGR-TK plots of other orders. So I'll pause here and see you in a next video.